And so, yeah, there is a need for independent tools for doing dengue on nanopore, and then also just for typing dengue as well.
All right, welcome everyone to another episode of the Bioinformatics Lab Podcast. I'm Kevin Libuit with Andrew Page. We're from Phylogenetic, and today we're going to be talking about dengue and the bioinformatics resources to perform that type of analysis. Andrew, curious your perspective and your experience on being able to analyze dengue genomes.
First of all, dengue, tiny little virus, you think SARS-CoV-2. SARS-CoV-2 is massive compared to dengue. Dengue is about, 10K. But it's an important pathogen, in more warmer climates than south. And it's something that we, obviously in the north where it's not a problem, we sometimes forget about. And it does cause, people to become very, very ill and in some cases worse. And the bioinformatics tools actually are quite poor for it, which is quite shocking and surprising. So, horrifyingly, most people, if you want to type dengue using bioinformatics tools, you have to use this kind of closed source website, right, called Genome Detective. And you have to upload your sample and then, into a web form and then click, and then it gives you, analysis later. And, obviously if you want premium access, you can pay for that, and it gives you more ability, like to upload multiple samples and to do things faster and prioritize, which is not really what we're used to in public health, and it's really, really bad. And whilst it is supported by a few universities, so many well-known universities, it's probably not a sustainable solution for public health, and it's more of an academic thing. So obviously, this being an issue, other people have tried to tackle it. Our own Anesh Menj, she went and for her PhD, she went and did some software to try and do typing with phylogenetic placement. And then, you've got other labs, is it Nathan Gruberger? Sorry.
Yeah, Grubal over there at Yale.
Yeah. And he's got Verdi Hill and Chris been working for him. And so what they've done actually is gone and made tools, open source tools, thank goodness, for actually typing dengue in a much more sustainable way. So actually dengue has 4 serogroups, and so 1 to 4, and then within that it's broken down further. But they've applied basically the pangolin typing scheme, like that we're familiar with SARS-CoV-2, so like B.1.2.3, and that kind of way of breaking things down, and they've got defined rules, and it's much easier and sustainable for typing overall, rather than this broad 1 to 4. And the reason it does actually vary quite a lot. But anyway, so they've got tools, but it's for Illumina, right? So you give it the raw, Illumina reads, does an assembly, picks the right reference, and then it goes and will type it for you. But obviously, if you have an assembly or a nanopore, then you're screwed. The only nanopore software out there is actually for made by a PhD student, and fair play to them or whatever. But unfortunately, it's not runnable by anyone outside the PhD student, because it's all kind of just one script, hard coded everything for their machine. And so, yeah, there is a need for independent tools for doing dengue on nanopore, and then also just for typing dengue as well. So this is not a solution, but more of a call to arms. It's just something we need to do. And the way typing is done currently is phylogenetic placement. So first of all, you work out which reference is closest. So there's 4 different references for the 4 groups. And then from there, you do your typing on top of that. And by basically putting stuff in a tree. Not that hard, but, it is nuanced. And it is important to know what is circulating and what is not circulating, so you can monitor, because different countries have different types circulating. So, say Colombia, where I was recently, have dengue 3 circulating. And so they've actually gone and made their own primer schemes just to focus on that pathogen or that particular type, because they know that's what's around. There are more generic panels, like for Amplicon sequencing, Sorry, I should take a step back and say that the way that you sequence dengue is actually either metagenomics. So, I don't know, you take someone's blood, you sequence everything, or you go and do amplicon sequencing in a similar manner to SARS-CoV-2. So you have tiles and you go and sequence those, and hopefully they all work and overlap. There is a typing scheme from the CDC, which is tiled and will do all 4 types, which is awesome. But then you can have differences within countries, circulating strains. Maybe they need a specialist type, and so you can use primal scheme then to go and build your own Amplicon panel and then test that. So it is a bit more difficult when you have custom panels, of Amplicons, because obviously testing becomes more of an issue. It's better to have one panel that lots of people use, because then obviously you test in depth. Anyway, sorry, I'm digressing here. Dengue, once you have your nanopore sequencing, and you can fit an entire virus nearly in a single read with nanopore, and when you tile it, you'll obviously be breaking it up into like fragments of 400 or whatnot, and so you get quite nice chunks. You don't need that many tiles, to be honest, not that many amplicons, and then you get some really nice beautiful genomes at the end, consensus sequences, and then you go on and do your typing by phylogenetic placement. So I guess what I'm saying is this is a neglected pathogen. We really need to come up with better tools and better ways, better standalone methods that are open source for doing this.
Well said. And as someone who is never worked with viruses until SARS-CoV-2, I think I kind of got a really skewed perspective of the resources for typing in the viral world, because obviously we have now at this point a pretty robust system of SARS-CoV-2 typing, and then maybe I got a little bit involved in influenza, and similarly so there's pretty standard methods there. But then whenever you look at these other, especially vector-borne pathogens that are impacting millions around the world, I'm kind of surprised at how limited the toolkit looks like for the bioinformatics in that space. Because even doing the phylogenetic placement, there's a bit of a manual review there. You got to see, okay, and it's somewhat subjective of, okay, how close to this clade? What if it's in between? Then you need to really have a subject matter expertise of like, okay, this variant at this loci, this is actually more indicative of clade 1 versus clade 2 or whatever it is. So I think that's worth highlighting. And there's folks doing exactly that, right? The Gruball Lab is the one I'm most familiar with, and some of their next strain builds. And at least they give you a tree to place on. And so you can see kind of clear clusters and maybe where your sample lies. But that takes so much effort individually to do it. Whereas I'm used to throwing it to next strain, throwing it to pangolin, and getting that kind of nomenclature out so that you could do the proper statistics on seeing the different circulating strains, and then even maybe the more granular phylogenetics based on a single clade, so you can get a little bit more understanding of the relatedness here. So yeah, it's always something surprising to me, just seeing how much more there is to do on all these other viruses that, as you mentioned, is largely neglected sometimes.
So there is an external build for dengue, but there's a lack of data out there. Because we get we're too used to the bacteria world where everything is just open by default, and WS, people just share their data. In the virus world, it's very different, and it's much harder to get data. People don't share it as much. They share it in closed archives, or they share restrictions, so it's not necessarily an NCBI and open. And so a lack of data is a huge issue, and it doesn't help. So putting stuff onto a tree in Nextstrain can be more of a challenge. Yeah, it's.
I was gonna go ahead. Sorry, finish that thought.
Yeah, no, it's just it is a totally different world, and then even typing, the virus world just don't use even the same nomenclature. I can't even say that word.
Nomenclature.
Yes. Thank you for saving me. They don't use the same things, and they'll call things wildly different names, or place names, or alpha, beta, gamma. It's crazy Wild West compared to bacteria, where it's like we've sorted this out 100 years ago.
Yeah, I was going to bring up that exact point. where we have a proper taxonomic system, which of course at the nuances at the edges, we all have our debates on, like, whenever a genus or species gets renamed and it's like, oh, okay, this is annoying, or this is, some really pedantic, of which I love, conversations that go on in the bacterial world. But at least there's a system and an orientation to the naming that we can at least agree on, and we even know when it changes, we can make those accommodations. Whereas in the viral world, it's largely still ad hoc a little bit, where it's like people are actively proposing new systems to clade, type, and provide monikers to what we're seeing at a genetic level. Like even pangolin, like that was SARS-CoV-2 is, hey, this is a proposed system by which we can make sense and mark and categorize the circulating variants we're seeing of SARS-CoV-2, and it sounds like similar efforts still need to be done to bolster our understanding and our communication really around dengue.
Yeah, absolutely. So watch this space. There's so much more to do in it, and hopefully people will, tackle it properly this time.
Yeah. All right. Awesome. So with that, we'll see everyone in the next one. And yeah, thanks for that discussion.